Hi, hello, my dear students. This is your Prasanna Harikrishna sir, Assistant Professor of Botany, SRR Government Arts and Science College, Karimnagar. So, in this session, we are going to see non-cyclic photophosphorylation. In the last session, I have given you the detailed, structure of non-cyclic photophosphorylation and what are the complexes which are there in the thylakoid membranes are discussed in my earlier session. So this session includes how the electrons move through different complexes in the thylakoid membranes. Clear? For example, so this is also called as Z scheme. This is also called as what? Z scheme. The word Z scheme was first used by Hill and Bendall. Hill and Bendall. So Hill and Bendall are the scientists who used the word Z scheme for non-cyclic photophosphorylation. So why it is called as Z scheme? Z scheme nothing but what? In non-cyclic photophosphorylations, electrons move from one complex to another complex in Z manner. Z manner, like the movement of electrons like this. So if you see Z in this manner, this complex 1 and this is the final electron receptor. So from here to here, or you can say A and this is B. So from here to here, electrons move in Z manner. Hence it is called as Z scheme. Okay. So the word Z scheme was used by Hill and Bendall. Clear? So where it occurs, for example, this is a chloroplast. Okay. Then these are the thylakoids. Okay. If you isolate one thylakoid, so it looks like this. Okay. This has different complexes, different photosystems. Photosystem 1, 2, like that. Okay. So if you cut this photosystem 1 and photosystem 2, if you see the diagrammatic representation, it looks like this. So this is a diagrammatic representation of non-cyclic photophosphorylation. Okay. So this side it is lumen, this is stroma. Okay. Stroma of chloroplast, lumen of chloroplast. Now this is PS2 or you can say P680. This is what? PS2. The active or reaction center of PS2 is P680. Then the light receptor will be this one. The light receptor is LHC. I have already told you the detailed information of LHC in my previous classes. So light harvesting complex 2. Okay. So 2 is associated with 2, PS2, photosystem 2. Clear? Then it receives sunlight. That is H nu. So H nu stands for sunlight. Okay. So whenever it receives sunlight, then the sunlight reaches to the reaction center of this entire complex, that is P680, that is chlorophyll A molecule. Then so it will be in excited condition and it loses electron. So this particular electron is received by Pho. So Pho stands for pheophytin. So this entire structure is discussed in my previous classes. So then electron move to the complex structure, that is PQ, plastoquinone. Then plastoquinone becomes PQH2, that is plastoquinol. Then plastoquinol again converted into PQ minus, that is semi-plastoquinone. Right. So whenever there is a conversion, two protons from stroma reaches to the lumen. So this is what? 2 protons. And there is a complex called OEC, that is oxygen evolving complex. So oxygen evolving complex is associated with photosystem 2. Okay, so where the water molecule is broken down into 2 protons plus 2 electrons plus half O2, means oxygen is formed. These two protons combines with 2 protons, 2 plus 2, 4 protons. Clear? Then there is another complex, that is cytochrome B6 and F. Cytochrome B6 and cytochrome F complex. This electron move to cytochrome B6 and reaches to cytochrome F. Then cytochrome F2, there is a mobile carrier nothing but it moves from one location to another location, that is PC, stands for plastocyanin. Okay, so finally this electron from plastocyanin reaches to PS1. This is PS1, means P700. This is PS1. Okay, so it finally electron reaches to PS1. So there is another light harvesting complex here. Okay, this is LHC1. So LHC1 nothing but light harvesting complex 1. So always 1 associated with 1, that is photosystem 1, light harvesting complex 1. Light harvesting complex 2 is associated with photosystem 2. Okay, then so it also, this particular photosystem also receives sunlight, right? So it also receives sunlight, that is H nu, and this sunlight reaches to P700, that is a reaction center. Then finally it lost electron. This electron is reached to iron sulfur protein. So iron sulfur protein from here to FD, ferredoxin. So ferredoxin is involved in the conversion of NADP into NADPH2 in stromocytes. Okay, then these 4 protons move to so these 4 protons move to oxysome. This is what? Oxysome. Right. So 4 protons are available now, but this oxysome allows 3 protons at a time. Okay, so now 3 protons move through the oxysome to the stroma. So whenever the transportation of proton takes place through the oxysome, there is a combination of ADP with PI and there is a formation of ATP. So this is all about what? Non-cyclic photophosphorylation. This entire picture was drawn in my last session, and all the complexes which are involved in the transportation, which are there inside the thylakoid membrane, are discussed in my last class. Okay, so today we are going to see how the electrons move and what the mechanism. Okay, so then come to first one, that is light harvesting complex 1. So start with here also, So LHC1, light harvesting complex 1. So light harvesting complex receives sunlight, right? So this is a sunlight, it is received by light harvesting complex 1. Then this light reaches to the reaction center. I have already told you what is meant by reaction center. Reaction center is nothing but a chlorophyll A molecule which is very active in the group of chlorophyll molecules. Okay, so this is chlorophyll A molecule of photosystem 1, that is considered as P700. P700 nothing but it is a chlorophyll A molecule and which is more active, and finally this light energy reaches to this chlorophyll A molecule, that is P700. Okay, so P700 is a chlorophyll molecule. Whenever it receives sunlight, then what about this is molecule, what happens after receiving energy or sunlight? This is the molecule, that is chlorophyll molecule. So there is an outer orbit, okay, outer orbit. So it is composed with electrons. Whenever it reaches sunlight, nothing but energy, the outer electrons are lost. So first of all, it will be in excited condition whenever it receives energy in the form of sunlight. Okay, so the exciting condition of P700 is denoted as, for example, this is what P700, I am writing here, P700 is denoted as P700 star. So star indicates exciting state. If there is no star over the 700, that is called as what? Ground state. This is ground state, this is excited state. Okay, so now this P700 will be in exciting state whenever it receives light energy from light harvesting complex. Okay, now this excited chlorophyll A molecule, that is P700, loses the electrons. Then these electrons reaches to the iron sulfur protein. Clear? This is a first electron receiver of PS1. Okay, so what is the first electron receiver of PS1? That is iron sulfur protein. Okay, so this is a first electron receiver. Whenever electron receives, when what is meant by oxidation? For example, oxidation means what? For example, addition of oxygen. Oxidation means what? Addition of oxygen, removal of protons or electrons. Okay, then what is meant by reduction? Reduction nothing but what? Addition of protons or electrons and removal of oxygen. So this can be called as what? Oxidation and reduction. So what happens here? Iron sulfur protein receives electrons. Okay, whenever the electrons or protons are received by some substance, that can be called as a reduction. Okay, so this iron sulfur protein get reduced after receiving electrons from P700 or chlorophyll A molecule. Okay, then this iron sulfur protein again loses the electron. These electrons are finally reached to ferredoxin. Okay, so this is an end receiver of electrons in PS1, that is photosystem 1. Okay, now this ferredoxin, ferredoxin involves in the formation or conversion of NADP to NADPH2. Whenever NADP is converted into NADPH2, two electrons are donated from ferredoxin. So first electron lost from the P700, first electron reaches to the iron sulfur protein, then iron sulfur protein to ferredoxin. And again sunlight is received by LHC, then goes to P700, one more electron is lost from the P700 and this electron reaches to iron sulfur protein, then finally ferredoxin. Means for every two electrons are added to NADP, then NADP is converted into NADPH2. Okay, so this is about complex 1, that is photosystem 1. Now this photosystem 1 lost two electrons, right? So two electrons are lost from this particular chlorophyll A molecule, means photosystem 1. Means there is a deficiency of electrons in the photosystem 1 because it lost two electrons. Every time whenever it receives energy, light energy, then there is a loss of electrons. If there is a loss of electrons, this gets deficient. Okay, electrons are deficient in photosystem. So it should be it should be substituted by another system. Okay, so there is a loss of electrons. We have to we have to donate some electrons from somewhere. Okay, now this is about PS1. Then come to PS2. So what happens in the PS2? So PS2, there is a light harvesting complex. It receives Hnu, that is light energy. Then this light energy is received by P680, that is a chlorophyll A molecule like P700. This is called as reaction center, which is very active chlorophyll A molecule. Then what happens? Same thing happens here also. The outer orbital electrons are lost. They will be in excited condition and they will be lost. Okay, so this electron is lost and electron is received by phaeophytin. Okay, so the first electron receiver of PS2 is phaeophytin. The first electron receiver of PS1 is iron sulfur protein. Okay, now this electron is donated to plastoquinone. So plastoquinone, after receiving electron, it becomes into plastoquinol, then semi-plastoquinone, then plastoquinone. Like electron transport system in respiration, there is ubiquinone in electron transport system. Like that, here plastoquinone E is there in the photosynthetic mechanism. Okay, so whenever there is cyclic manner of plastoquinone, the two protons from stroma gets entered into lumen sites. Two protons move from stroma to lumen. Okay, now remember, so this electron from plastoquinone, electron is moved to cytochrome B6F. How the cytochrome B6F made up of, I have already told you in my previous session. Then electrons are reached to plastocyanin. This is a copper containing electron receiver. Then this plastocyanin donates electrons to PS1. Okay, what happens here? First of all, PS1 loses electrons and there is a deficiency of electrons. That deficiency is substituted by whom? PS2. So PS1 deficiency of electrons are substituted by PS2. So this is movement of electrons. It looks like what? So when you draw the diagrammatic representation, so this looks like Z. It looks like what? Z. Okay, so hence Hill and Bendall called non-cyclic photophosphorylation as Z scheme. Clear? So this is about all electron movement of non-cyclic photophosphorylation. Now, so here at PS1 there is a complex called oxygen evolving complex. So oxygen evolving complex involves in the production or creation of oxygen from water. So water is more abundant in the lumen. So hence lumen is considered as reservoir for water. So this water gets broken down into two protons, two electrons and oxygen molecule. Okay, oxygen gas. Then for this conversion, Mn+2 and Cl-. So manganese and chloride ions are required. They are essential for the conversion of water into protons, electrons and oxygen. This entire process was put forth by Robin Hill. So whenever there is an availability of HNU, that is sunlight, this water gets broken down into protons and oxygen through oxygen evolving complex. This entire concept can be called as Hill reaction, or you can say photolysis of water. I have already told you, you can see in my previous classes. This is what? Photolysis of water or Hill reaction. Okay, so these two protons combines with the two protons from stroma. So 2 + 2 4, these four protons move towards the oxysome. That is the fifth complex oxysome, or you can say coupling factor. Okay, but this oxysome cannot allow 4 protons at once. It allows only 3 protons at once. Clear? Now, according to chemi-osmotic theory, chemi-osmotic hypothesis, I have already told you during electron transport system in the respiration. So Peter Mitchell in the year 1961 proposed a new theory or new hypothesis that is called as what? Chemi-osmotic hypothesis. This tells about proton motive force, PMF. Proton motive force. So whenever non-cyclic photophosphorylation takes place, two protons, for every time two protons come from stroma to lumen, and there is a formation of two protons from the water. So every time these protons move from stroma to lumen. Protons move from stroma to lumen, and there are no formation of protons from the water. Hence, the proton number increases. Proton number increases in the lumen. Proton number decreases in the stroma. Okay, proton number increases in the lumen side, proton number decreases in the stroma side. Okay, so this is major difference or major osmotic proton gradients variation or difference you can see in both the sides. There is less protons available in the stroma side, there is more protons available in lumen side. So what happens after some time? So more protons move from higher concentration to lower concentration. Okay, so more concentration is with lumen, less concentration is with stroma. Then according to Peter Mitchell, that is chemiosmotic hypothesis, protons generally move from higher concentration to lower concentration. This particular movement takes place through the oxysome or F0 F1 complex, or you can say coupling factor. Okay, then for every time 3 protons move through the oxysome. So whenever 3 protons move through the oxysome, nothing but lumen to stroma, there is a combination of adenosine diphosphate with phosphate, and there will be formation of ATP, adenosine triphosphate. Okay, so this is all about the different kinds of movements in non-cyclic photophosphorylation. Then tell me, what are the outputs or end products of this particular non-cyclic photophosphorylation? See, oxygen is an end product, NADP is an end product, and ATP is an end product. Remember, so 3 compounds. What are those compounds? Oxygen, ATP, NADPH2. So these 3 compounds are formed as end products during non-cyclic photophosphorylation. Clear? Oxygen is evolved, oxygen is formed where? PS2 only. Oxygen does not form at PS1. So PS1 does not involve in the formation or creation of oxygen, whereas PS2, photosystem 2, involves in the formation or creation of oxygen. Because why it forms? There is oxygen evolving complex which is associated with PS2 only. Oxygen evolving complex is not associated with PS1. Okay, so this is all about non-cyclic photophosphorylation. So thank you one and all. In next session we will see cyclic photophosphorylation. Thank you.